Hello, and welcome to Living Seeds, the missing microbial piece. I'm Michael Washburn, the Seed Savers Exchange Preservation Director, and I'll be your host for this session. I want to send a special thanks to Chelsea Green Publishing for sponsoring this session. Now, today we're here with Matt Powers. He's an author, educator, entrepreneur, farmer, seed saver, citizen science, and family guy. Matt Powers is the author of Regenerative Soil and the Permaculture Student Series. Today, author and educator Matt Powers will be teaching you how to inoculate seeds with beneficial biology, learn from the past, and connect it all to current regenerative soil science. Now, according to Matt, you will always inoculate your seeds once you understand the behaviors of endophytes and the process of rhizophagy. Matt, thanks so much for being here.
Thanks for having me. This is going to be fun.
All right.
Hey, everybody. Thank you for being here. I'm excited to talk about this topic. I started with seeds, many years ago. Many folks know that about me. And so to come full circle is going to be a lot of fun. All right. So today we're talking about living seeds, the missing microbial piece. Thank you for being here and taking the time to think about seeds, to congregate with people who love seeds. Do you know about horizontal gene transfer? And do you know that it's more significant than evolution in adaptation? So, what is it really, right? It's horizontal gene transfer. That means it's consumption. They're consuming genetic material from the world around them. Conjugation, it sounds exactly what it is. And then viruses. And viruses like mu viruses are exchanging information and genetic material between bacteria. Conjugation, the bacteria literally can connect, and then they look like little rabbits spinning and doing their thing. And they're exchanging DNA in milliseconds between each other. So why is this relevant? I thought this was only proven with E. coli, Matt. Well, E. coli is the fundamental feedback loop for all life on Earth. So there's millions of species, but only a handful are pathogens. They're found in your soil, in your water, and on all surfaces. And they're actually plant endophytes. Did you guys know that? The reason they're in the animal digestion to begin with is because they were in the plants to begin with. They're in 18 to 20% of all soil and compost samples I've ever DNA tested. And when we say E. coli, it's like saying animal or mammal. There's a big difference between your dog, a chipmunk, and a shark. Or a hippo. Hippo's the most dangerous of all. So there are different. There's a handful of pathogens, and there's millions of species of E. coli. 20 to 40% of the soil is unclassified DNA. So that's absorbed in horizontal gene transfer. DNA can take up to a million years to decompose in nature. So that's why 20 to 40% of the DNA that we find in the soil is from this heritage of life. We look at organic matter aggregates. Where did that organic matter come from? Living organisms. And that means that it's made of decomposing living organisms, meaning plants and animals, and that you know what I mean? That's genetic material. Literally, that's genetic material. So most people don't think about that, but that's what it is. Horizontal gene transfer means that E. coli sources new genes from its environment constantly. So when you have anaerobic conditions and you have anaerobes show up, this is how. There's E. coli in your gut because E. coli was in the plants to begin with. And when we look closely at plants, we look at the endophytes and the phloem, it looks like this. These are fungal endophytes here. And so do you see how the xylem's on one side and the phloem's on the other? We're saying it's in the phloem. You see how it's on one side there? Look, it's on one side. Pretty amazing when you get to actually see these things. This is mycorrhizal inoculation of a corn root. So why is this important, right? Because we are constantly absorbing new information about the world around us and adapting at a microbiological level. That has direct influence over our genetic expression. So We are always taking in new information. The food molecules and nutrients influence and trigger epigenetic expression. Microbes digest our food for us. Like, let's just be real. We are just like plants are relying upon microbes to get our nutrition. Are your soils and plants inoculated with the right biology for your needs? All right, well, before I get any further, who am I? Well, I'm Matt Powers. I used to be a musician. I became a public school teacher. I was a sub and then a public school English teacher. I started teaching permaculture. And then over time became a citizen scientist because of the demands of both my students and delving deeper and deeper into soil science and composting and permaculture. My family has grown up with me doing this, and I in the past 8 years, over 8 years now, I've released over 24 books, over a dozen online courses, and it's all in an effort to make a bridge to the regenerative future. So adults to teens to even K through 12 can actually have the tools to live regeneratively, to start regenerative entrepreneurial businesses, and to have success in those endeavors, because there are proven paths, because they're based upon regenerative cycles that are implicit in the natural world around us that we just need to harmonize with, and then we get abundance and growth that is superior to anything that we have currently in the grocery store and in mainstream farming. So we're seeing incredible things, and we'll talk about that. Regenerative seed equals the best seed. So what is best? Well, let's look for these attributes: the volume of fruit, leaf, and seed, the resiliency, the drought tolerance, the vigor, the season length, how long is it going to stay alive, and the best of the best. Who's the best of the best in this category, in this family, in this group? And then utility. How useful is this?
Hey, Matt,
Yeah.
may I interrupt real quick?
Yeah.
I apologize, but we're only still seeing the first slide.
Oh, so our efforts were not successful.
Now I saw you change. You're on your corn slide.
Okay.
There we go.
Yeah. There's a lot of pictures here I want you guys to see. So we're going
Yep.
to go through it all again in review in hyperdrive.
Yeah, let's do that. Sounds
All right.
great.
So we now know what horizontal gene transfer is, and it's more significant than an evolution. So the genes adapt in real time because microbes, they're the canaries in the coal mine. Even though we say they have the most effect on pH and EH and nutrient absorption, they're actually here. That's better. Let's make sure we get the full thing. They're actually the most affected and first affected when change happens. So if you're in contact with the feedback loops of life, whether it's E. coli or like rhizobia passes through the ruminant digestion. And we have to understand that there are indicator species of everything, whether pathogens or beneficials, but there are dominances as well. And when we look at endophytes, beer yeast, Saccharomyces cerevisiae is right up there as well, along with E. coli. But these microbes are incredibly important. They're endophytes, and endophytes fix nitrogen internally. So these plants are so dependent upon these microbes. And it feels weird because you're like, E. coli, the news told me that was bad. And the reality is, they don't know anything on the news. I mean, come on. They're just reading off a piece of paper. So you can really see right here, this root cross section here, the endophytes are in the phloem. And you can really see it's on one side, right? Do you see how it's xylem and phloem? One side is the phloem, and you can see it. That's fungal inoculation. That's epifluorescence lighting. That's the work I do. And this is the corn seedling root, and you can see the inoculation rate. You can see the growth. So why is this all important? Well, because we are in direct relationship with the world around us, and it affects our genes in real time because we are microbially fed. through the food and the microbes inside the food, and everything triggers our genes because microbes digest our food for us, just like they do for the plants. So if your stuff, whatever it is, your soil or your plants aren't inoculated with the correct biology, we're going to fail in a variety of ways to provide what could have been. So who am I? I'm Matt Powers. I was a musician in New York City and in LA. My wife got cancer and we moved out west. I became a school teacher. And then over time, started writing books and I became a citizen scientist. My family has grown up with me writing all these crazy books and launching all these courses and transforming. And so this has been my last 10 years of my life, and I live it, I breathe it, I love it, and all of it's in an effort to make that bridge to the regenerative future, so that future generations can have access to ways to live beneficially with the world around them, with their community. They can start businesses easily as well. The entrepreneurship side of this is extremely potent because we can make better food, better medicine, and we can do it ethically. And so we can just blow them out of the water with transparency. I really value this. I was a high school teacher. I was teaching sophomores, so it's all about launching them into their future, and I'm still doing it just for adults and all the way through K through 12. So regenerative seed, it's the best seed. But what's best? Let's look at certain attributes: the volume of the fruit, the leaf, and seed, the resiliency, the drought tolerance, as well as the vigor. How fast is it growing? How strong is its stem? Seasonal length. Is it a long season crop? And is it the best of the best? Is it clearly the one that is better than all the others that you planted? And is it useful? Is this something that you really want to scale up and focus on, And then you want to select seeds from those heavy producers. You want to save, plants with the largest overall yields. They're going to bring you the most pounds per square foot of food, right? You're going to select seeds from your best plants in the worst soil and or conditions. You're going to select seed from your fastest growing, most vigorous plants. You're going to select seed from your plants that are first to fruits. So first fruits we let go to seed. And then that makes them more and more and more precocious, except you're now planting out those seeds at a greater number next year. And so the first one out of that group, it will be even more precocious. Select the best seeds from the seeds you saved, the heaviest, those without blemish, the most symmetrical, the darkest coloring, the largest, all of the ones with significance in different ways. And this can be subjective. You might be like, I like the speckles. We're collecting the ones with the speckles. And that's okay. This is part of it. Beauty is part of it. Multi-purpose seeds. I am from the permaculture survival kind of world. So edible seed, edible leaves, edible microgreens, edible stock. If everything goes wrong, we have food. And then drought tolerance. If suddenly we don't have any water, these seeds, this amaranth can grow without any water in that heat. I mean, look at the white ground on the left side. Oh my word. That is 140 degree soil. So you thought I was making it up, didn't you? Look closely. We can zoom. Let's zoom. Look. All right. So now you get it. And then, but even when you water, remember, you got to flush that water if that's been sitting in the hose all day. But look, mulch. Mulch is our friend. But that other section didn't have mulch. I literally just threw it on the ground. So I like to save seed. I like to grow really beautiful, really powerful food. Vigor, utility, and high yield. You want to look for that. You want to see expressions that feel inspiring, because that's what our plants should do and should be. So you were like, thought I was making fun of someone for selecting for speckles. I am that person. All right. So this is my Peruvian adaptation. So I adapted this to North America. This was considered impossible, but I did something that a farmer would never do. I put it on the afternoon side of a hill so the sun didn't hit it directly until the afternoon. Then I put it behind a tree so it was like super shaded, and it thought it was home, even though it was at 2,000 feet. And so it grew 16 feet high. It was wild. I needed a ladder, and some of them formed crowns on top of corn. And in a circle, a crown of corn, and in the center was the tassel. Unbelievable, just wild stuff, right? And that's all documented. So season length, earliest salon. That's actually in a book called Corn, Moccasin Roots, Shoots, and I can't remember the last word, but it's by Stephen Smith. And then thinking about season length even more, getting an early crop in there, getting the earliest crop in there, and then developing that early crop to be even earlier by doing things with the soil and permaculture and all those kinds of things will allow the soil to heat up earlier. So how do you get seed like all this? Well, start with heirlooms. And and remember, heirlooms to your area would be what the indigenous people, the native peoples grew. So that is always going to be the most vigorous seeds. So doing some research, learning about your place, learning about the history of that place. And this may be like iterations, you know what I mean? Because some of these places have changed. Some of these places no longer get the water they once did. You may have to look a little bit closer to the equator. You might have to look an analog that's drier. You might have to adapt, where you're buying your heirlooms from because of all those reasons. But look at those seeds first, and then start working on the analogs, and then start working on the things that you love, that you want to grow, that are expensive, and you want to save money and grow something better than you ever could get in a store. So scrutinize your plants and seeds and select carefully. Sow your seeds outside directly on the soil as much as possible. If you have bird pressure, you can cover things you can rake things in, but you could also cover things with like a netting or a shade cloth because it's tighter weave. There's a bunch of things you can do, or you can get like a Jang cedar and do it very shallowly, planting exactly precisely shallowly, and have it patted down really well, because you can do that with a lot of fine seeds that it's like difficult to hit that perfect when we're doing it by hand or throw sowing it. We want it to be tucked in, but Jang cedar is great for that. Water your plants as little as possible. Time plantings to the rain. This creates really vigorous, resilient seed. If you soak your seed beforehand, this makes this process much easier because they have moisture with them. They don't suck it out of their environment in order to do what they need to do. So sow is broadcasting. And then we save seed aggressively and end up with only the heaviest seeds. So all the light seeds, all the smaller, less developed seeds, they get blown away. And then we dry farm as much as possible. I'm growing Sonoran dry farm beans right now. And you breed your own vegetable varieties. So you can adapt things, you can change things, you can create new varieties of plants. It's absolutely incredible. So check that book out. I believe the sponsors for this episode of the Seed Savers Exchange put out a new edition of this book. so yeah, no, that's just, I didn't realize Chelsea Green was going to sponsor us, but this is Carol Depp's part of their team. And her books are my favorite books, probably by Chelsea Green. So thank you guys for providing a lot of my favorite books. But there's more to this, okay? How do plants actually feed? Let's get down to the deep education. This is where I like to go. So pH, the hydrogen cycle, power of hydrogen is pH cycle. Okay, keep that in mind because look, hydrogen protons, H plus, hydroxide, OH. Did you know that is literally the pH scale? Acidity is measured by how much protons you have, right? And then alkalinity is measured by how much hydroxide you have. So plants are literally determining the pH. And they're also dealing with energy. And notice that when you're adding nitrates and any form of nitrogen that you add to oxidized soils turns into nitrate within hours. So it requires 4 times as much water. So it's really important to recognize that when plants are healthy, when plants are in neutral or acidic soils, they're adding energy to the soil, they're adding carbon to the soil. But when they're doing this, when they're stressed, when they're being fed nitrates, and they are oxidizing the soil. So we have to recognize this is a fundamental process. When they release those protons, they displace the nutrients because they flood it with these protons, and so that's how it happens. It's not like they're actually attracting the K plus with the H plus. No, they're displacing it. And then there's the soil food web that everyone knows and loves. Everyone knows and loves soil food web. This is the most accurate soil food web map diagram that's available that shows you the relationships with the arrows. Each of those arrows is peer-reviewed and researched and cited. So, and this is typically what we were told about, the soil food web. It's cakes and cookies, bacteria and fungi feed upon it, protozoa feed upon them, nematodes feed upon them, and everyone else. And then the manures from the protozoa and nematodes, those make organic matter and feed the plant roots. So that's typically what we're told and what's been passed around and has developed into an assumption. But that's not all. There's more to the story. It's going to get pretty wild here, so hang on. And then we know rhizobia. That's another form of rhizospheric relationships and synergy and symbiotic relationship. And then there's bradyrhizobia. This is the one that shares nitrogen horizontally before the plant dies out. This is the one that harbors it, and then the plant has to break down to release the nitrogen. This whole confusion. This is what Elaine Ingham was talking about years ago when she talked about bradyrhizobia. People thought she was talking about this, but they're all in the rhizobia family. So anyway, it's really important to know all like the details to really understand a lot of these things that experts have said over the years. Mycorrhizal fungi. This is what arbuscular mycorrhizal fungi really looks like. It's really important to recognize that it doesn't look like the trees when you put undisturbed roots, undoctored roots, unchemically altered roots. The thing is the arbuscular shape, that tree shape that everyone sees and recognizes is actually an artifact of the test. And so they only have that image for a few seconds and then it's destroyed. And so they're burnt nubs of a much more delicate and interesting process. And epifluorescence microscopy is showing it to us. And then the manual lighting technique as well. That's something I was able to pioneer. And so we can see the endophytes inside of this is pumpkin stem hairs, those white spikes. There's bacteria and fungi inside them. You won't see it with light, with just bright field light microscopy. You need the epifluorescence. And then epifluorescence also shows us, fungally inoculated stomata. The red there is actually photosynthetic chlorophyll. We can examine leaves. We can examine the trichomes, which are the hairs on leaves. And the thing is, most of the nitrogen taken in from plants when they're working properly is from the air. And so when we think about the fact that 78% of the air is nitrogen, of course plants have a relationship with them. So there's more to this. So have you guys heard of rhizophagy yet? Well, I'm here to tell you about it. So roots eat the microbes alive. They farm them at the root tip with exudates, but exudates are reabsorbed. Up to 90% of exudation is reabsorbed. So it's like in and out, breathing, waves on a beach. And they suck in the microbes that have been growing on the exudation and then bombard them with superoxide and destroy their outer cell walls. And most of them expire. Some of them regrow these cell walls as they form the root hairs. And in fact, rhizophagy is the reason root hairs form. And so they reform their cell walls as they are in the root hairs and then are expelled from the root hair tips. This is really well documented, and it's seen in all plants. It starts from the moment of birth, and it is the pathway that mycorrhizal fungi, hijacks. And it's also the real power of what compost tea actually does. Compost tea provides plants already charged biology so that they get sucked in and then they drop what they were carrying because they're bombarded and they leak their electrolytes, they leak their nutrients. So it's they reabsorb 90%. This is not cakes and cookies. If this is cakes and cookies, this is Hansel and Gretel story. So that whole idea, and it's also a shotgun blast. It is not a conversation that's specific. Plants aren't like expending the energy to be like, I need Septiglamus deserticola. Hello, Septiglamus. They're not sending out specific signals or specific exudates. The exudates, they can't control. The thing is, the exudation is part of photosynthesis, and they just release it. And whether it's good or bad depends on the health of the plant. And if it's bad, it calls in the bad microbes to come and digest it. If it's good, it calls the good microbes to come protect it. So, and then once the microbes say, hey, I'm here and I'm infecting you, the plant reacts. And then basically these microbes a lot of the time are using the same pathways that viruses and pathogens use, and they are simply beneficial. So you'll find a group of pathogens and then one of them isn't. One of them's a beneficial, and you're like, well, that's really weird. As a spirillum's a spirilla, but like, if you look at, what Elaine Ingham said, she said spirilla was bad. It's like, yeah, but this is the exception. And so it's really important to recognize that there are exceptions in every group, in every family. In my work, I go down to the individual, not just the morphological. I look at the actual DNA in the individuals at work, because if we're dealing with nutrients and specific plants and endophytes and internals, and you need to know the specifics of who's who, not just fungi isn't just fungi. And that's been the big problem. And rhizophagy, when endophytes are absorbed, like the E. coli, like beer yeast, beer yeast and wine and bread yeast was already inside all the plants and on the surfaces of the plants too. So it was there. It was going to happen. It was going to happen naturally because it was always there. So when these microbes are inside the plant, though, they get bombarded with the same superoxide and they have to release nitrogen. And that nitrogen provides the plant with another form of nitrogen that they wouldn't be, that's independent of the soil and the air, and it's internal. It's already there, perfectly in situ. And if you think about it, the yeast feeds upon sugars. And if the plant has too many simple sugars, it gets overloaded and then gets attacked by pests. These yeasts help sop up those extra sugars. And then nitrogen fixing microbes that are also endophytes, they literally can transform the nitrogen to other forms of nitrogen or provide nitrogen from different forms that are there. Provide other, you know what I mean? They can transfer the nitrogen that's there. And there's air that's taken into plants. Remember, some plants have air pockets within them. So all of these things are happening at the same time, and depending on the plant, maybe one of those things is happening, and depending on the microbes, maybe some of them are happening, but could happen. So it really depends on the plant we're talking about. And the endophyte we're talking about. That's why regenerative soil, my book, goes down to the specifics of all of that. One of the things I really wanted to do was see this. So I grew out pumpkin seedlings. I fed them microbes and EM, and they were really healthy. And then I put them under the microscope with blue methylene, or methylene blue, excuse me, and stained it. And you can see the microbes inside and the ones being expelled quite clearly. It's pretty wild, all right? We can verify the science of regenerative soil. That's been my work, and it's amazing how there's so many people with PhDs that have never used a microscope but talk all day long about soil. They've never actually gone and verified the things that they teach. I care about this so much, being able to be transparent, to showcase how we test things. This is why I'm going to create that R soil database. Here's another example. You can see it coming out of several pores. Plants are active. It's true. But they're not the ones controlling everything. They can't shut off the sun. They can't stop photosynthesis. They can't stop releasing exudates, because exudates are a combination of secretion and excretion. They can't control it. So they're active, but they're also passive. They're lit up by the light. So they have no control. They're going to just start again, even if that morning comes, that dawn comes, and they haven't processed yesterday's sugars and nitrogen. They're just going to back up, and that's why they're going to become overloaded with simple sugars or nitrates, and you'll get aphids, so feeding on the sugars, and you'll have things like grasshoppers feeding on the nitrates and sucking caterpillars feeding on the nitrates. They're passive and they're reactive. So they get some pest damage, but if they have the tools, they'll react. If you feed them properly, they react. If you add the microbiology, they react. Microbes are being digested. They're being stripped and restored. They're being forced to fix nitrogen. I mean, it's as Dr. White says, you fix nitrogen or die inside the root. And they're being partnered with. There's symbiotic relationships left and right. And they're also being beneficially coexisted with. There are things existing in the soil that are doing their work, releasing, whether it's microbial secretions or whether it's a byproduct of them doing their work and then them being consumed by other members of the soil food web. The benefit of coexistence in a diverse environment is always hand over fist greater when there's coexistence. Plants rely upon biology for their immune systems. The plant health pyramid. So when I learned about this, I became very, very curious, because the top two levels of plant health were described by John Kemp as requiring robust biology. And it's like, well, what does that mean? And he never gets to specifics. And so I I've gone that's like I mean, this is what regenerative soil was all about. It was about finding out who is who and not just settling at good fungi, good bacteria, and generalizing like that because that's in the light of what I know now, that is so just blah. It doesn't mean anything. I mean, the fact that saprophytic fungi is what was being measured for years in compost. Meanwhile, saprophytic fungi, other than a very few that become endophytes, and I'm talking about very few, things that are random, like turkey tails and endophyte, kind of cool, but your compost isn't like turkey tail heaven, right? So we have to keep these things in mind that the saprophytes are not going to directly benefit your plants. And not only that, the saprophytes are the ones that are in general in the much more alkaline environment. So you have the hot compost that's pH 8. Typically, it's pH 8 and maybe have beautiful fungi in it, but dang, that's not going to help your plants in Lockout Central, bringing things up to pH 8. And then light microscopy and all the things that people are looking at in Brightfield that traditionally people are looking at until my book and my courses was they couldn't even see the fungi that was actually important to plants. They couldn't see arbuscular mycorrhizal fungi, which partners with over 90% of plants. I think it's over 95% of plants. And you look at mycorrhizal fungi, it's like 98, 99% of plants. The weeds and brassica can partner. It's typically non-mycorrhizal, but with trichoderma's help, it can partner with arbuscular mycorrhizal fungi. But our amaranth, our Chenopodium family, our beets, our Swiss chard, they're non-mycorrhizal, and they're going to release hydroxide, and they're going to keep soil alkaline and oxidized and not let it. I mean, the organic matter over time will build up and force things down, make your amaranth shrink each season, but it fights it. So it's really important to know who we're talking about so that we can achieve that top level. We need those endophytes. We need them inside. We need to verify these things. We need to inoculate things because we are relying upon these plants. And we are all reliant upon the microbes for our digestion, the wild animals too. This is why it's such a crime that fungicides were used all over America and all over the world. But these forests grew up as strangers to each other because there was no mycorrhizal network connecting them all. And because of acid rain, sulfuric acid is a fungicide. So we've through those decades of acid rain, we sterilize the environment of fungi to a great degree. If you went up to New Hampshire in the 90s, late 90s when I was growing up, you'd see the exposed roots of all the trees in the forest, and it just was so bad that they had they had stuff that was like copper, and I think I don't know what they were doing. They were trying to bond up the sulfur with copper and other things in situ, and they had these little balls they were dropping all over the forest. I don't know if you guys remember this, but man, it was terrible. It was a terrible solution. And getting rid of acid rain ended that, of course, but it also exposed how badly we were farming as well and how deficient our soils were in sulfur. So it was really incredibly important for us to see that, digest that, and recognize that. But the wild is in deep need of biology to return. And that's one of the hardest things for people to accept and understand, because the wild is the wild. If we just left alone, it'd be fine. Matt, just left alone, it'd be fine. But no, it's not. No. So we have to recognize that we have the power and responsibility to re-inoculate, to partner with microbes, to see beyond the surface level of the world around us. Do you want to partner with biology? Because your seeds do. So let's get some practical stuff in there, right? Make this practical. Let's get some how-to. So the root, leaf, and soil inoculation, We want to we want to give the microbes back to the plants. These plants all evolved with these microbes in place, and so they need them. And that's the real reason we haven't seen the regenerative response that we typically would see in many places. Landscapes are becoming more brittle. Plants are becoming weaker. And it's because they're lacking their partners. They're lacking the powerhouse and the upper levels of their immune systems. I collect compost and watch compost as it ages, and I inevitably get these gnats because I'm in an older house for this lab. This is an older building, and so I'm constantly having these bugs dive bomb me as I'm talking. So forgive me if I'm like, wow. So foliar feeding, root inoculation, seed inoculation, and soil soaking. We are going to bring the biology to the plant. We're not going to overwhelm the plant with biology, but we're going to consistently bring the biology back, because if your soil is dead, if your soil is simplified, if your soil lacks organic matter, if your soil leaches easily, if your soil is oxidized or alkaline, it's not going to keep the biology. So you can inoculate, you can do all these different things, and you'll have flushes of reaction. But they're flushes. They come and go. So I want you to take that on board, that, and then all at once. I think I hinted at it earlier. If we had something phosphorus rich when we're inoculating with mycorrhizal inoculant, we're going to inhibit the mycorrhizal growth. So we need to actually have strategy. We need to test in time, apply strategy, look at the cycles of nature, understand the nature of our plants and the microbes themselves, so that nature is what's informing our decisions. This is the most important thing as an educator, is to, let the text speak for itself. Let the student understand and perceive, because that's the highest level of understanding, is fluency, you becoming the teacher and teaching. And the only way, like, I teach fluency. That's like what I do. And so we need to get out there. We need to do these things. People are coming up with new ways of doing this all the time, because they're thinking about things from the perspective of the plants. So water, EM, mycorrhizal inoculants, and other biofertilizers can be added in any of these junctures. And this is what plant cells actually look like. They look like pieces of glass. Well, because they're little greenhouses that absorb sunlight and turn it into energy. And this is what happens. And when they're fungally inoculated, they glow characteristically. And when the roots are fungally inoculated, they do as well. We can use EM, or we can brew our own, because if you look, it's lactobacillus, right? Well, the ingredients, water and molasses. And so if you're doing water kefir, you'll have the yeast, you'll have the lactobacillus. And then you can add Rhodosodomonas palustris, which is the third ingredient in EM Pro, which is incredibly important. And these yeast flakes are characteristic. So Rhodosodomonas palustris can be added in, and they advertise it as it being there, but as you just saw, there's no guarantee that it's there. It's something found in all composts to varying amounts. It's found in water, mud, soil. It feeds 4 different ways. It feeds on light, heat, sugars. It's wild, and it can take in electrons. It can feed on carbon sources. It's amazing. And they're facultative anaerobes, and so they can live in the space between true anaerobic and true aerobic. And remember, if it's true aerobic, that would be like us tilling constantly. And so natural soils are actually facultative anaerobic. They're not anaerobic and they're not aerobic. They're facultative, which is in between. Just like there are microaerophilic ciliates. Very important. There's a lot of debunking I've done in my recent book. So the key ingredient in EM is this microbe, and you can buy this microbe at Algae Barn, purple non-sulfur bacteria plus Rhodocytomenalis palustris. They advertise it as PNS. Algae Barn online. It's a pure culture. It's wildly beautiful, and this is the key ingredient after yeast and lab in the EM consortium. So in other words, you can just work with those because the EM is a trademark name for selling those microbes. The microbes themselves are not trademarked. So it's just something that everyone's come to say, and it's a common way of talking about these things, but they're totally just beer yeast, the kombucha and water kefir lactobacilli, and then this aquarium supply common ingredient, Rhodocytomenalis palustris for controlling algae. So this is what it looks like. And so my new book has keys so we can compare and identify microbes. It's beautiful. It's not purple, it's red. But that's the reality. And when you look it up online, there's only one other person that has any images of these. It's pretty wild, actually. So I was really happy to get these images and to be able to verify using their images, these images, and the Bergey's manual to verify this, because there's so few examples of this. And so I was, it's pretty wild. This is an incredibly rare image that we're looking at. So we were talking about Saccharomyces cerevisiae, beer, bread yeast, right? It's found in EM because plants get more nitrogen and phosphorus when it's in their roots. It's a biofertilizer, improves plant vigor. It does not produce protease, so it cannot use nitrates. And that's why lab is so effective in partnership with it, because that turns nitrates into amino acids. It can feed on a variety of sugars and utilize urea and ammonia. So this is incredibly useful. And if you've added, NPK fertilizer, whether it's natural or not, you actually can see the microbes using this technique. So like if you added bird guano or bat guano or something to an EM extension, you can actually, when you test it 2 weeks later and say, oh, there's no there's no nitrogen and no phosphorus in this liquid now. Well, let's put it under the microscope and look, and then suddenly all the glowing ones are the ones with phosphorus, and they're all plump looking. They're happy because they're full of food. They're full of nitrogen and they're full of phosphorus. So it's important to use the tools that we have. This is from my new book, Regenerative Soil Microscopy, that's coming out soon. All these microbe images and profiles are from that book. So lactobacilli and all they're rods. It's very, very easy to identify. So it's a trademarked name and their trademark product that they sell. The microbes themselves are not trademarked. Test your compost before using it. Know what you're using. Partner with microbes. If things are high in phosphorus, they inhibit the mycorrhizal fungi. If it's high in nitrogen, means we need to be careful with how we apply it. Also means that maybe we need to do more work to make it ready. But it also means it's good compost, because if there's no nitrogen, then there's no nutrient cycling, because their manure is nitrogenous, right? So then regular feeding, you want to do foliar or soil soak, weekly or spoon fed. And then you also want to add those microbes when you're drying down as the same way. And then when you're soaking seeds, you're really actually priming them. So when you're drying down, you're adding microbes on that will die, but some will survive in the skin and the rind of the seed, in the seed coat. And then soaking seeds is a way to bring it all back, a way to bring that microbiology back. And so we're priming them as well, because we're adding the moisture, they're waking up, they're getting the microbes and nutrients right there in real time with rhizophagy. When we dry down seeds, it kills some microbes. Not all, but most. When we restart the process, everything goes in. So EM, mycorrhizal inoculants, and other biofertilizers, they're all going to go in. And these are specific biofertilizers. They're all specifically can work together. Like azospiralium works really well with arbuscular mycorrhizal fungi, for instance. There's synergetic partnerships between bacteria and fungi that people don't even know about. I mapped them out in Regenerative Soil, the first book in the trilogy. And we continue to do so in the new book, Regenerative Soil Microscopy, which is the second in the trilogy. So we get specific because fungal saprophytes are good for composting, but unless we're talking about turkey tail, it's endophytes and mycorrhizal fungi that actually matter because those are the partners for plants. And mycorrhizal fungi will increase the root surface area by 10,000 times. And they're way better at solubilizing phosphorus than roots are. And there are specific groups. There are a few overlaps with saprophytes, right? But mostly they're not endophytes. Mycorrhizal fungi, yeah, we just said that. Roots feed on rhizospheric bacteria through rhizophagy. Half or more of certain nutrients being acquired are gotten through endophytes. So you can have up to 45% of the CO2 provided for photosynthesis to plants and coming to them internally from the microbes. You can have the same, 45%, nitrogen. Those kinds of numbers are what people are seeing in tests. But what can we expect with biologically inoculated seed? Well, they're regenerative seed. So you would have the same results as regenerative soil, especially if you've done the work of regenerative soil, created your holistic soil management plan and implemented it. So this is all just a component of regenerative soil, the soil biology, the organic matter, the air and water, the plant roots, photosynthesis, and minerals. Having the right seeds with the right biology makes it so that the plant roots and photosynthesis happen properly, and relationships with biology and the creation of organic matter increases exponentially. So that means pest infection, virus, and disease immunity. That means high nutrient density becomes possible. It means faster maturation rates and higher yields. And we've seen this in John Kemp's work. We've seen this in Chris Trump's work. We've seen this in my work. I took a corn variety, and if you know corn days, I moved one variety 30 days. So made it more precocious. And seed saving does this, but also regenerative soil, mineral coherence, the right biology. It's not one thing, it's all things. Increased secondary metabolites and aromatics. So these are going to be medicinal qualities, the things that we love, taste, and recognize as what makes food enjoyable. And that is all microbes. And then a massive decrease in fertilizer inputs. Farmers are just tiptoeing into this, and they're seeing huge reactions. People are adding EM in with their regular synthetic nitrogen fertilizer and Haber-Bosch, process, and they're suddenly increasing their organic matter levels by 0.5% in 2 to 5 months in Arizona desert farming, right? And it literally, that is such a game changer. That is so much faster than anything else that's been documented. So we can change fertilizers now using biology. We can make them safer for the soil. We can then make it so stable that they can step out further, because now there's enough organic matter. Now there's enough life. Now there's enough nitrogen fixation happening in real time in the plants and in the soil. Taper that down. And that becomes less work over time. Less inputs means less spending over time. And so you earn more. You have more life, more and free time with your family, or more time to expand those gardens if that's what you would like to do, because that's what I always do. So if you'd like to learn more, please check out regenerativesoilscience.com. There's courses, books, free resources, and more. Reviews from people you know and love, like Permaculture's David Holmgren, Dr. James F. White. Just the Jeff Lowenfels, the list goes on and on. Thank you for being here. Thank you for hanging out with me. I'm Matt Powers. Grow abundantly, learn daily, and live regeneratively. And visit us and learn more about regenerative soil science.
Well, Matt, thank you. Man, that's awesome. You ran through all of it. Unfortunately, I'm not going to have time for questions. We ran a little bit over, but I wasn't about to stop that awesome train. That was not going to happen. So, yeah, thank you so much for this. I think a lot of people got some just some really good stepping points to go ahead and really dig deep into all the work that you do. Because you've got some great publications out there, and just and one of my favorite things is, yeah, that good old compost tea. Boy, I put it up against so many things, and yeah, I'm right there with you. It is just fantastic. So again, Matt, thank you so much for putting all this together and sharing with this community. I know everybody appreciates it, and yeah, I look forward to seeing you again. So thank you so
Thank
much,
you.
Matt. All right,
Have a great
thanks
one,
everyone.
everyone.
Bye now.
Bye.